Check, check. Check, check. Check, check. Hey, this is the docking AI, and these are the CASP results. This is our first prediction here that we're making. We're making it for T1105V1. Here's what the docking looks like. Wonder if you can hear my cat. And so this is showing the active site here. And we'll just hide the surface. I'll just show the side chains to make it simpler. And there's that cysteine that was mutated. The hydrogen bonding doesn't look super great. It's nice that that's packed there. So there's this tryptophan here. And another tryptophan there, and a tyrosine, so it's hydrophobic. Are there any? This does have the hydrogens on there. Wonder if there's a simple way to just show the donors and acceptors. That's a nice interaction there. Hi, hi. Oh, my cat's gaming when I talk. Hi, hi. Love you, Shobai. Okay, we should just go back here just to make sure. And this is the ligand. MDL format. And that should be today that it's expiring. Quinine salvage enzyme. I did start around the 9th. No, around the 6th, so I've had a while, but it's taking a while. I didn't get much done, actually. But I did use Nina. Basically just used Nina. Now we're just using this collab fold notebook. Very kindly provided by the Nina folks. And we did do a little bit different. We got it from our GitHub, so you can just get it from there. That's just like open source there, target and initial, and then we use that as the ligand. We used that to just get us find where that active site was, and then get us a better docking, and then we did this as well. We added these flex dist ligand, and we use that ligand, and then 3.5 angstroms, randomly chosen. It's about the length of like maximum length. You should start looking for like any kind of hydrogen bonding interestingness. But I also saw 4 angstroms. Hi, bye. And then we also outputted those flexible residues, and then we joined them then later too. We'll see how that does. It's better than nothing. At least we got something in. Got something in at least. That's what counts. Let's get it. Bye. And then I guess the thing we'd want to do is like do like a sanity check on that. And just look at like, use like our big brains and use like science. Duff 2419. And we saw that here. PDB. This one. We've looked at this. We've looked at this a lot before. So, Duff 2419. Structural characterization of Duff 2419, a quinine salvage protein by Shi Hao Hong, Sana Al-Kuzwani, and Manal A. Swearjo. Sorry for butchering your names. I'll try to get better. Quinine is a modified nucleobase. So here's quinine. This is quinine here. So it kind of looks like DNA a little bit. Oh, where are we going? It is biosynthesized de novo via 7-deazoguanine pathway in many bacteria and archaea. It is salvaged by eukaryotes where it acts as a precursor to the modified nucleoside equiosine found at the anticodon wobble base of cytoplasmic and mitochondrial tRNAs that incorporate tyrosine, aspartic acid, asparagine, and histidine amino acids. Check me up on those, but I think I get it pretty close. In human, quinine is exclusively retrieved from the diet and microbiome and is subsequently inserted in human tRNA by the human tRNA guanine transglycosylase enzyme. Woo! Good morning. That'll wake up your wow. I need a little tea break. Tiny smoke break. So, quinine is the only micronutrient that directly affects efficiency and accuracy of translation of mammalian proteins, with roles in metabolism and brain health and aging. It is required for the biosynthesis of BH4, a vital cofactor for numerous enzymes in the body, including those involved in the formation of the amino acid tyrosine and the key neurotransmitters dopamine and serotonin. Formation, it is BH4. What's BH4? Tetrahydrobiopterin, BH4. Okay, well, that doesn't look so different. Quinoid dihydrobiopterin. Oh. So we're right down in all those things. Interesting. Recently, okay, that's a little too crazy. Recently, DUF2419, encoded by C9ORF649 in human, was identified as a protein family involved in quinine salvage in plants, animals, fungi, and even in some bacteria lacking the 7-deazoguanine de novo biosynthesis. Nevertheless, the exact biochemical function of DUF2419 remains unknown. So we don't know what it does actually. DUF2419 is deleted in cases in acute myeloid leukemia, and Q-modified tRNA levels correlate with tumor aggression, implying a link between Q metabolism and cancer in humans. Oh. Here we report the X-ray crystal structure of DUF2419 from Sphaerobacter thermophilus, and it's approximately 37 kilodaltons, determined at 2.1 angstroms resolution by selenium-based multi-wavelength anomalous dispersion methods. MAD. The structure reveals that's the acronym for it, is MAD. The structure, it's a super funny story. Multi-wavelength anomalous dispersion. It's so cool, actually. Who is it? Like Henderson? Who is that? Yes, Hendrickson. Oh, Wayne Hendrickson, exactly. MAD, abbreviated MAD. It's super cool. And here's SAD. And it helps to solve the phase problem because there's certain atoms that diffract electrons, diffract the photons anomalously. That diffract the X-ray photons anomalously. And you can use that to solve the phase problem. Wayne Hendrickson. Okay, back to here. The structure reveals a monomeric protein, so just like one protein. We're looking at that here. Oh. Monomeric protein. We could show that with the cartoon. (331 residues, so not too big, not too small. The structure reveals a monomeric protein belonging to the orthogonal helix bundle fold. Whoops, excuse me. Three helix bundles are among the smallest and fastest known cooperative folding structural domains. Three helix bundle in the villain headpiece domain is only 36 amino acids long and is a common subject of study in molecular dynamics, because its microsecond scale folding time is within the time scales accessible to simulation. Fascinating. Cool. There's all about that. Okay, getting too deep again. A DALI search for similar structures returned several 8-oxoguanine DNA glycosylases, which cleave oxidized guanines in DNA. And now this is probably jumping too far ahead, but we can look at our DALI results here. And let's just look at the first few. Now, the query here isn't the crystal structure. The query is the one from the PDB. Oh yeah, see there's that pocket. The query here is from the PDB. And then these are the most similar ones returned by DALI. If you haven't used DALI before, it's super amazing. Query color. Oh, just make it a rainbow and then see. Oh, actually, this monochrome is better. That the overall structure of those helices is conserved, and then it has this thing in here, and that's in 1XQP, and that is this one, complex with PA-AGOG, whatever that is. Oh, okay. That's just another big one then. 3D view. Structure. Oh yeah, that looks similar, right? You got this guy here, and then this ribose here. That's the weird thing. Ours has that. The other ones stop there. And then ours has this wacky thing that comes back over. I wanted to look at that as well. Oh, that's actually better. Oh, then you see the backbone as well, and the backbone will definitely be. Now that, I don't know about that leucine over there. That all seems really unhappy. Interesting. But yeah, I'm using Nina. Journal of Chem Informatics, Nina 1.0, Clinton Docking with Deep Learning, David Ryan Coase, Andrew McNutt, Paul Frank Kerr, Risha Agarwal, Tomohide Masuda, Rocco Maley, Matthew Ragoza, Jocelyn Sanseri, David Ryan Coase. Cool. Oh, sorry. Wow. And lots more too. Cool. You can see that I already have looked all that up before. Cool, computer science. Oh, Camacho. That is so familiar. Protein. Yes. As you can see, go to all this. Carlos Camacho. Farmer. Cool. Cool. What was CSAR 2011? Structure activity, well, that's QSAR, CSAR, interesting. But we all know that CSAR, the QSAR is incorrect, so. And this is all open source as well. It's all just here. I'm just trying to learn, just trying to help. Okay, cool. Okay, 11:17. Do we want to continue? Okay, let's continue that paper. Where were we? Got lost in the paper. This one. Okay. Let's move all of these out somewhere further. Okay. DALI search, we showed that. Structure-based alignment reveals 22 sequence similarity and only 2 invariant residues. The catalytic asp residue and the lyse essential for recognition and positioning of the substrate during the base hydrolysis reaction. Base hydrolysis, so what's that? Queosine base hydrolysis. What's that? Queosine. No, this is super useless. DNA base hydrolysis. Oh yes, what was that? That was the it found the oxoguanine DNA glycosylase. How does that work? Okay, so now we need to find this one. Okay, let's look at this one. Just look at the first one. Oh, there you go. Okay. PDB. Sad. Oh, it's just that. Oh, it's just that. Okay, well, that doesn't matter. Structure. Yes, structural insights. Oh, yes, cool. Oh, wow. Oh, cool. What's this one? Ooh, there you go. There you go. Okay. See, I wish you could just do that in the PyMol right there. Oh, see, that's so nice. Okay. Oh. Oh, what's going on? Oh, that is it. That is it. Okay, is the sugar towards that? Or is that cysteine? Spheres. Okay, so that's at cysteine. But then what? It shouldn't have a cysteine. That's a mutation. Yeah, I think that's a little too... Okay, we have to read that more carefully. See, should have that all in like a machine learning thing so we can get all that. Okay, we should just like read this paper more carefully. think we jumped ahead too much. Yeah, that's with the DNA. Well, we also did have all those other ones. Why don't we just load those all in? File, initialize everything. File, open. And now we go here and okay, we'll go, first of all, we'll go to great. Okay, and then let's open Okay. Now then, these are quite different. Like this whole thing here, label. That. What if we look at the collab fold? This one. I see like all of that. See, maybe I should have just like cut all that off, because that's kind of really just going right in the active site there, right after that tyrosine arginine. I wonder how that would would have affected the... Let's see, I did that. I guess we'll all be doing that. What is that? Is that on purpose or not? And it's unsure about that. That's... It looks pretty well the same. Like, it would have been, if I had had more time, I would have gone and looked at those other be good to just have a way to do that really quickly, but just like do it slowly too. Cyclopentyl ring of queen. Cyclopentyl quiescine. Let's look at that again. This one, what was that? Cyclopentine. Cyclopentyl. That's that one. What did they say about it? Cyclopentyl. Okay, now where did we put it? So let's keep hide everything. The surface. Hide the cartoon. Show lines. It looks pretty good. There's like space in there. But you don't have anything, that everything is unknown. I don't know, that one looks pretty good to me. That's just everything, and let's just make sure. Open. Casp, and we'll do all people. Cool. Good to look at all those structures. It's too bad I could just like load them. It's like a little bit of a pain to do it in PyMol. I should just like write some scripts to visualize all that. Well, lovely audience, I think I'm gonna end the stream. And could have submitted more, but I don't really see what the point is since we're not testing our own stuff. There's a lot of things to test. We're mostly just testing the system. Thank you very much for watching. This has been SNIS.